Good day, everyone, and we are going to report about the polysaccharides. So here's the table of contents of our report. 1. The major types of starch. 2. The process of starch gelatinization. 3. The process of stalling. 4. Modified starches and the other polysaccharides. And last is the references. So I am Olive Magdalene A. Jandog together with Angelo Delight to report about the major types of starch. So first, what is polysaccharide? Polysaccharide or polycarbohydrates are the most abundant carbohydrate found in food. It has a long chain polymeric carbohydrates composed of monosaccharide units bound together by glycosidic bonds. The commonly found monomer units in polysaccharides are glucose, fructose, mannose, and galactose, which are the simple sugars. Next is the structure of polysaccharide. Polysaccharides are complex biomacromolecules that are made up chains of monosaccharide, and the bonds that form these chains are glycosidic bonds. Polysaccharides tend to be a long molecules consisting of many simple monosaccharides or monomers. Two monosaccharides may undergo a condensation reaction in which two free hydroxyl groups react to form a glycosidic bond, and a molecule of water is eliminated. As such, polysaccharides tend to be a large and nonpolar, and therefore do not dissolve in polar solvents such as water. Most of polysaccharides consist of 6 carbon monosaccharides, resulting in a formula of C6H10O5. There are three main functions of polysaccharides. One is to provide structural support. Its linear molecules link cellulose and chitin, which are strong and rigid. Wherein cellulose is the primary support molecule in plants, while fungi and insects rely on chitin. Number 2, sending cellular communication signals. Polysaccharides are used for sending cellular communication, are often covalently bonded to lipids or proteins, forming glycoconjugates. And last is the storing energy. Polysaccharides are used for storing energy. It tends to be branched and folded upon themselves. Examples of storage polysaccharides are starch in plants and glycogen in animals. So, aside from glycogen and cellulose as an examples of polysaccharides, starch is also one of it. So, what is starch? Starch or amylum is a white, granular, organic chemical that is produced by all green plants. Starch is manufactured in the green leaves of plants from excess glucose produced during photosynthesis and serves the plant as a reserve food supply. Starch is stored in chloroplast in the form of granules and in such storage organs as the roots of the cassava plant, the tuber of the potato, the stem pith of sago, and the seeds of corn, wheat, and rice. Starch is a complex carbohydrate that exists in many foods, including grains, vegetables, and fruits. Worldwide, it is the most common carbohydrate in human diets and is contained in large amounts in staple foods such as wheat, potatoes, maize, rice, tapioca, and cassava. Next is the structure of starch. Starch is a natural polymer or polysaccharide, meaning it is a long chain comprising one type of molecule. It is made up of long chains of glucose molecules that are bonded together through covalent bonds called glycosidic bonds. Starch is created by covalently bonding a chain of glucose molecules and have a basic chemical formula of C6H10O5, where N is the number of glucose molecules in the chain. It is a polysaccharide comprising glucose monomers joined in alpha 1,4 linkages. Starch is made up of long chains of sugar molecules that are connected together. The linear polymer amylose is the most basic form of starch, while amylopectin is the branch form. There are four major types of starch, and first is the amylopectin. Amylopectin is a non-crystallizable form of starch and a water-soluble polysaccharide and highly branched polymer of alpha-glucose units found in plants. It is one of the two components of starch, the other being amylose. Amylopectin is a large organic molecule usually produced in plant cells that contain repeated units of glucose molecules. Amylopectin is stored in plants as temporary granules that can be later broken down by enzymes to release the glucose as it is needed for energy production. Both plants and animals can break down amylopectin into glucose to use as a source of energy. Next is the structure of amylopectin. Amylopectin is a highly branched polymer formed by many glucose units linked together. It is a polymer of several D-glucose molecules. And 80% of amylopectin is present in starch molecule and has a properties that amylopectin is known to be soluble in water. Amylopectin molecules are linked by alpha 1,4 glycosidic bonds between two glucose units in the straight chain and alpha 1,6 glycosidic bonds at the branching. Each amylopectin molecule may contain 100,000 to 200,000 glucose units, and each branch is about 20 or 30 glucose units in length. So, these molecules are bushy and nearly spherical in shape. Next is the iodine test. The iodine test is a starch identification test based on a chemical reaction. Iodine and starch generate a unique purple blue black colored complex in color. In this test, and a negative result, which means there are no there are no starch present, was the yellow color of the iodine solution. In an iodine test, the helical structure of amylopectin is disrupted by the branching of the chain. So instead of a deep blue-violet color, amylose gives with iodine. Amylopectin produces a less intense of reddish-brown color. So that's all for my part. Thank you for listening.